Hi. How's everyone doing tonight? Is everyone ready to learn some science? Who'd like to make $500 by participating in science? Great, lots of interest. Well, I am Natalie and I'm an immunologist. And despite my aspirations to one day be an evil scientist, I'm not one of those I'm not one of those lucky few who got that sweet Pfizer vaccine money in the pandemic. I work at Samari, also known as the giant cheese grater next to the hospital. And I'm here today to tell you about my research. Partly because you might find it interesting, partly because I'm a narcissist, and partly because I need participants for my new study. So in our lab, we study the immune system, and specifically we study vaccines, how they work, how our bodies respond to them, and importantly, how to get that dose of autism just right. So usually when we're assessing vaccines, we're measuring vaccine responses. And what are these? Well, normally we're measuring the production of antibodies, which are immune proteins that circulate in the blood and bind to disease-causing germs to prevent further infection. And also T cells, which are specialised immune cells that can directly hunt out and kill infected cells to prevent further infection. Now, a good vaccine following vaccination will ideally induce both of these immune responses. But unfortunately, following vaccination, not every person is equally protected from disease. So to demonstrate this, I'd like to introduce you to Bert and Ernie. This tall drink of water on the left is Bert. Bert has amazing vaccine responses. After he gets vaccinated, his antibodies are blazing. His T cell responses are on fleek. You can tell just by looking at him, this man knows how to mount an immune response. He's got big energy. 10 out of 10 would bang. After vaccination, Bert is likely to be protected from disease. There you go, Bert. On the right, though, we have Ernie. Following vaccination, Ernie's antibodies may as well be absent. His T cells are basically on strike. And, well, basically, Ernie has the immune system of a sickly child from a Charles Dickens novel. Following vaccination, Ernie is not likely to be protected from disease. Boo, Ernie. Now, in a lot of cases, following vaccination, we don't know why some people, or most people, might respond like Bert, and some people respond like Ernie, or some people respond somewhere in between. What makes a Bert or an Ernie? Is it Bert's haircut, or maybe like the vertical stripes? Is it because of Ernie's childhood trauma? Well, in our lab, we're investigating the reasons why vaccine responses differ between individuals, to hopefully find correlates, targets that we can exploit to improve poor immune responses in those that need help. Now, there are a lot of things that can actually influence your response to a vaccination, things like age, or genetics, or past history of infections. Unfortunately, these are all things that we can't change, but we are focused on one factor that we can change, and that's the gut microbiota. The gut microbiota are the billions of organisms present in your intestines, and they help digest your food every day. These include bacteria, also viruses, funguses, and some parasites. Now, we've known for a long time as scientists that the gut microbiota is really important for immune responses. But work in our group has also shown that when we look at vaccine responses following vaccination in babies, that babies that have good vaccine responses have certain kinds of good bacteria that are lacking in babies that have poor vaccine responses. And similarly, using animal studies, we've shown that vaccines don't work as well in mice that were previously fed antibiotics, which depletes the gut microbiota, or mice that have an absent gut microbiota altogether. Now, this is all well and good, but our studies so far have a major caveat. We're not able to show causation in humans. We've been able to show correlation between different microbes and vaccine responses, but we can't show causative evidence that this works in humans yet. And what kind of evil scientist would I be if I didn't test in humans? So to test this, my team and myself have established the Antibiotics and Vaccine Immune Responses Study. Yeah, that's a really catchy name, I like it too. In this study, we're going to take healthy individuals under 35 and divide them into two groups randomly. One of those groups receives no antibiotics, while the other group will receive a short 3-day course of antibiotics. And following that, everybody gets vaccinated with one of the OGs of the vaccine world, the BCG vaccine. Now, we don't really use BCG routinely in Australia anymore, but it has been used for hundreds of years around the world to protect against tuberculosis or TB. It is actually a bit of a crap vaccine though. It only protects about half the time. But this is really good for us for assessing factors that could lead to poor immune responses. So following vaccination in both of these groups, we're going to take blood samples to measure the level of antibodies and T cells, like I mentioned before, and see which of these participants look more like Bert or look more like Ernie. And now here comes the fun part. To assess the gut microbiome, I need to measure some stools. And I don't mean a trip to IKEA. I mean assessing the bacterial content and type in a load of fecal samples. So to do this in our study, we're going to get participants to provide fecal samples to us. We send them a little stool kit, which again is not something that you get from IKEA. And in that, it's going to have a little tube with some special science liquid inside, which will preserve the stool sample for analysis and inactivate it so it's not infectious, for us to analyse it in the lab. And one of the easiest ways for me to receive the sample in the lab at that point is through the mail. Yeah, I know, I didn't know this was legal either. But I checked with Australia Post, you can totally do this. In fact, you can even do this online. There's like a load of places you can go to send your poop to someone. So for $25 in shipping, you can send that poop straight through the mail, which is insane because we're paying people to do it. This poop sending business has gotten so out of hand, even the Australian government is getting in on the action. So how's our study going so far? We started recruiting participants late last year, and so far most of our participants have something in common. They're all female. The feedback that we're hearing from men is they're not really comfortable providing the stool sample. Apparently men have a huge problem touching butts, which is totally at odds with my husband's search history. He's there, by the way. And other available evidence following my research indicates that a lot of men have no problem with butts whatsoever. So we need to find ways to get men more involved in our study as well. So what are we hoping to get out at the end of this? We're hoping that following our vaccination of our participants and assessment of immune responses, that we can show that participants who had antibiotics have reduced or weaker immune responses compared to participants that did not receive antibiotics. And then by assessing their stool samples, we can identify which microbes could be responsible for this discrepancy. And this could be really helpful for us to design interventions that we could give to at-risk individuals to improve their vaccine responses. Things like dietary changes, or probiotics, or other targeted medications that improve the gut microbiome to turn these unfortunate Ernies into beefy birds. So we need lots more participants in our study, both male and female, to help find out how your poop can help make vaccines better for everybody. And I guess what I'm trying to say is, if you do have an urge to send your poop through the mail, can you just think of me? Please enjoy my shameless plug, and thank you very much for listening to my talk.